Welcome everyone to Kinexum's webcast on targeting metabolism to attack obesity, diabetes, NASH, CVD, dyslipidemia, and aging itself by Michael Zemel, PhD, founder and CSO of NuSirt Biopharma. A couple housekeeping details first. While you're listening to the presentation, please place your microphones on mute so we can optimize the sound quality for everyone. Second, if you are only calling in but have a chance to log in via the internet, I would urge you to log in so you can see the slides Michael will be sharing and talking to. Third, an audio recording and a separate file containing Michael's slide deck will be posted on our Kinexum website, kinexum.com, probably the other side of the weekend. And fourth, if you have questions, please enter them in the chat function of the Uber Conference platform we're using. You can open it up by clicking the cartoon comment bubble icon in the right column of your screen, and Michael will address them at the end of his prepared remarks to the extent time allows. Just for a little background, NuSirt's chairman, Joe Cook Jr., a former Lilly executive and former CEO of Amylin and Ironwood Pharmaceuticals, was a speaker at our inaugural Metabesity Congress in London last October. Metabesity is a term coined by Kinexum's executive chairman, Zan Fleming, to refer to a constellation of chronic, non-communicable diseases and conditions, such as those mentioned in the topic of this presentation, that may have common roots and therefore may be susceptible to common solutions. NuSirt's approach is a paradigmatic example of pharmacological targeting of metabolism, which seems to have a cluster of beneficial metabesic effects. This is fascinating work, and I turn the mic over to Zan Fleming to introduce the speaker.
Well, I did want to have the privilege of giving an introduction to our speaker, Michael, who I have known for over 10 years now, and what a delight that relationship has been. Michael is such a creative thinker, but importantly, he reduces thoughts to practice and ultimately does experiments and shows very exciting results, all in a nutshell. The exciting thing to me about what Michael is doing is he's getting to one or more products that could be a means of preventing disease, not just managing it. And also related to the concept of metabesity, he may also be developing a product that could modulate the aging process itself, that is, to increase healthy lifespan. So for all these reasons, it's a great pleasure to introduce Michael, and won't take any more of the time. We could go in all day about the fun that we've had together, Michael, on the road, but take it away.
Zan, thank you very much. I very much appreciate the opportunity to present what we've been working on, and I am going to focus this on targeting energy sensing systems, and I'm going to jump right in. And hopefully everyone who's online has accepted the invitation to share the screen and is able to see the slides. What I've shown here is a rather conventional view of a cluster of diseases that we might call common diseases of nutrition. And okay.
All right, could we ask others to mute their lines? We're hearing some interference in Michael's presentation. Thank you.
Thank you. So this rather conventional view on this slide simply points out that we have a cluster of conditions that enjoy, if you will, a relationship. There is either a unidirectional influence, or in some cases a bidirectional influence. In the case of obesity and diabetes, for example, you have a bidirectional influence, in some cases unidirectional influence. But we are looking at these as distinct and separate diseases. And just to go through a quick example, a less common example, this slide shows a fibrotic liver, or a cartoon of a fibrotic liver in a NASH patient. And through a variety of signaling molecules that we don't have time to discuss, there's the opportunity to raise blood pressure. Hypertension, in turn, because of its hemodynamic effects on the liver, does exacerbate development of fibrosis in a NASH patient. So you have this bidirectional influence, and yet we recognize each of these two diseases as separate entities, and we're not pointing to a common pathophysiology. I'd like to progress us from that to a different perspective. Using Zan's term of metabesity, I'd like to take a metabesity perspective, and this will be the lens for the rest of this discussion. And that is that impaired energy regulation at its core can be expressed in multiple pathophysiological manners, but that there is a core defect which can be expressed as excess energy storage, as impaired insulin sensitivity, as increased inflammation, impaired vasodilation, et cetera, so that we wind up with these very common diseases of aging and overnutrition. And based on the individual and perhaps concomitant modulating factors, which one or ones are expressed in any given individual may vary. So I'm going to be focusing primarily on two energy sensing systems or nutrient sensing systems, those being AMPK and SIRT1. And both of these, both independently and together, and there is a bidirectional interaction between the two, as shown on this slide, have the ability to modulate each of the conditions that I showed you in the previous box: lipid metabolism, insulin sensitivity, energy metabolism, inflammatory and oxidative stress, and vasodilation for that matter, and thereby have an influencing effect on obesity and the other diseases of aging. Just to put a fine point on it, we know that both AMPK and SIRT1 are suppressed with age. They are suppressed in conditions of overnutrition as well as physical inactivity, and that in experimental systems, suppression of each or both of these systems result in diabetes, hypertension, dyslipidemia, obesity, atherosclerosis, fatty liver disease, etc. So let's take a more careful look. We're going to discuss SIRT1 and AMPK as fuel sensors. They do have some other functions, but we're going to focus on their ability to sense fuel. In the case of AMPK, we are sensing an abundance of AMP relative to ATP. And in the case of SIRT1, we are sensing an abundance of NAD relative to NADH. So in both cases, what we're sensing is a low fuel gauge. In both of those cases, we have upregulation of both of these enzymes, which together activate catabolic systems to increase energy supply while putting the brakes on anabolic systems that require more energy, such as protein synthesis and fatty acid synthesis. So in this slide, we're taking a simplistic view of how these two enzymes simply affect obesity and energy disposition. And what we see is by activating SIRT1 and AMPK, and I know I have eNOS on these slides, we're going to talk about it a little bit later, we have an effect in adipose tissue of suppressing lipid synthesis and increasing mitochondrial biogenesis. And in skeletal muscle, we have a very similar effect, increasing lipid oxidation and mitochondrial biogenesis. And the net effect is essentially a transfer of energy from storage in adipose tissue to oxidation in skeletal muscle, with obviously beneficial effects in obesity, and we will see in other disease states as well in a moment. Much of the rest of my talk is going to focus on activating this system through a combination of the amino acid leucine and some additional drugs. We have focused on leucine by demonstrating that it lowers the activation energy for SIRT1, enabling activation at ambient levels of NAD. In other words, non-starvation or non-fasting levels of NAD, which sets the stage for the enzyme to be further activated by other drugs. So when we combine leucine with other drugs, we are able to essentially generate a caloric restriction mimetic or a fasting mimetic without actually fasting. Now, before I go any further, those of you who are familiar with leucine are going to want to raise a question. Leucine is known by many as a stimulator of anabolic metabolism through mTOR, the target of rapamycin. And what I want to point out to you before we go any further is that takes a lot of leucine in both animal studies and some clinical studies, as well as in vitro, on the order of 2 millimolar. Now, to give you a frame of reference, that's approximately 20 times the fasting level of leucine, which is about a tenth of a millimolar. You might push it up to a quarter of a millimolar with a good steak dinner. The levels of leucine that we are using take you a little higher to about a half millimolar, and we've shown that we get the predominance of a catabolic effect, stimulation of SIRT1 and AMPK activity, and actually putting the brake on mTOR at these concentrations. So we are dealing with the catabolic side of leucine, even though it has the capability of doing both. So from this, we've generated a therapeutic approach of combining leucine with co-activating compounds to activate SIRT1. Leucine by itself does have an activation, but it's not sufficient to be a potent drug. We need co-activation. So we combine leucine with co-activation to activate SIRT1 and thereby also activate AMPK, and again, affect this same cluster that you've seen on the previous slides. So here is a preclinical sort of proof of concept. This is a study that we did a few years ago in diet-induced obese mice, these animals were fed either a control diet or resveratrol, a recognized SIRT1 activator, although it takes high concentrations to do it. And we tried both low and high concentrations. We combined leucine with resveratrol or the leucine metabolite, shown here as HMB, with resveratrol. And what we showed is that we could, in doing so, increase muscle glucose uptake quite substantially. And here you see the PET image lit up with either the leucine resveratrol or leucine HMB combination compared to the control mouse. This was associated with weight loss. This was associated with reductions in circulating insulin and corresponding increases in HOMA. And this was at concentrations of resveratrol that exerted no significant independent effect in these animals. And you see the corresponding changes in SIRT1 activity in the lower quadrant of the slide. And you can see leucine resveratrol and leucine HMB, even at low concentrations of resveratrol, resveratrol HMB, exerted comparable effects on SIRT1 activity as well as in vivo effects. So taking this concept to the clinic, we combined either leucine or HMB with low doses of resveratrol. And we're talking about low doses, 50 milligrams of resveratrol in combination with either leucine or the leucine metabolism HMB or hydroxymethylbutyrate. And what you can see here is that in a glucose tolerance test, we have an improvement with both combinations in both glucose area under the curve, insulin area under the curve, corresponding improvements in 60-minute glucose and 60-minute insulin. So we have a proof of concept here that the mouse data translates from mouse to human. It enables biological activation of resveratrol at low concentrations, but I think you'll agree that the effects I just showed you are modest. Now, those effects were in pre-diabetics. It's entirely possible that greater utility could have emerged if we had used diabetics rather than pre-diabetics. I cannot answer that question. But this left us wanting a more potent sirtuin activation or overall activation of the SIRT1 AMPK system. And what we did was we relied on our preclinical data, which showed that leucine could also amplify metformin action. Now, metformin is a known activator of AMPK, although it has some non-AMPK actions as well, related actions as well. Less well known is that metformin also has effects on SIRT1, indirect effects on SIRT1, which are very much amplified, as you can see here, in the presence of leucine. In our preclinical work, we showed that combining leucine with metformin enabled a substantial dose reduction, 83% dose reduction, and still yielding comparable efficacy, or about a 2/3 dose reduction to actually get superior efficacy to full dose metformin. So we said, let's test this. Let's do another proof of concept study. And we did a proof of concept study in diabetic subjects. These are data from a 28-day study. And what you can see is that from a fasting plasma glucose perspective, a low dose metformin, 500 milligrams or 1,000 milligrams per day, was equivalent to 850 milligrams metformin by itself in the absence of leucine given twice a day. So 1,000 versus 1,700. And likewise, if you look at CGM analysis, 24-hour glucose, you see again that the 500 milligram BID metformin combined with leucine was equally effective to 850 milligrams metformin given by itself. And you can see the magnitude of the decrease here. They were quite substantial, 50 to 60 milligram per deciliter magnitude. So this told us again that the preclinical data translates. Leucine clearly augmented the effectiveness of metformin, enabling activity at low dose, but it was a modest overall effect size, making us believe that if we want to really have a major effect on overall cardiometabolic health, rather than just modestly reducing the metformin dose, we might need an additional sirtuin pathway activator. And so what we did is we relied on the fact that there is another sensor. People don't often think of endothelial nitric oxide synthase, or eNOS, as an energy sensor, but it very much interacts with SIRT1. And in fact, there is a bidirectional relationship in which SIRT1 activates eNOS as the resulting nitric oxide has a positive effect on SIRT1 as well. So we thought, let's use sildenafil. Now, sildenafil is best known, of course, for its PDE5 inhibitory activity, but at low dose, sildenafil also has a modest activating effect on eNOS, a direct activating effect. And this is at concentrations that are much lower than those necessary for PDE5 inhibition. So we generated a triple combination of leucine, metformin, and as you see here, very low doses of sildenafil, either a 0.5 milligram or 1 milligram. We ran preclinical studies which showed that this combination reduced body weight, overall adipose tissue mass, circulating lipids, reversed fibrosis, inflammation, and steatosis in a mouse model of NASH, and that this triple combination was more effective than combining leucine with metformin without sildenafil, or leucine with sildenafil without metformin. So we took this into a clinical trial. We took 70 obese subjects and randomized them into the 3 groups shown here: placebo, low dose, and when I refer to low and high, I'm only referring to the dose of sildenafil. Leucine and metformin were kept constant, and so low dose refers to a 0.5 milligram of sildenafil. High dose, and you can put high in quotation marks, refers to 1 milligram of sildenafil given twice a day. And what you can see here is that in the overall cohort, the triple combination, dose responsively, here's placebo, here's low dose, here's the high dose, promoted weight loss. And it did so in a rather linear fashion, showing no signs of a plateau. And here you see the overall weight loss over the 4-month period. It was a 16-week study. Now, in subcohorts, this shows a subcohort of subjects that had another aberration, that is hypertriglyceridemia. We saw a more substantial weight loss of about 5 kilograms over the 16 weeks. We looked at other subcohorts as well. For example, we looked at a subcohort of individuals who were both obese and had elevated liver enzymes, as well as high liver fat. That was half the subjects, and those subjects also had a profound weight loss for this duration, given that there was no energy restriction or lifestyle modification. Those subjects lost about 4 kilograms. There was a corresponding reduction in blood pressure. So you can see about a 5.5 millimeter reduction in diastolic blood pressure in the full cohort, a less impressive drop in systolic pressure in the full cohort. However, if we look at those subjects who had hypertension coming into the study, and that was half of the subjects, you see nice reductions in both diastolic and systolic blood pressure. On a placebo-adjusted basis, this is about a 10 millimeter mercury drop in diastolic blood pressure and a comparable drop in systolic blood pressure amongst the hypertensive subjects. There was also a drop in hemoglobin A1C. Now, this is a very modest drop in A1C, but I want to point out that these are non-diabetic subjects. The A1C at baseline averaged 5.7%. And I also want to point out that here, the triple combination, here there was no dose responsiveness, leading us to believe that this A1C drop, consistent with our previous study in diabetic subjects, was probably driven by the leucine metformin component. We also see a very nice reduction in circulating triglycerides. Now, we saw this in all subjects, but it was most pronounced in those with hypertriglyceridemia, where we saw a very pronounced drop of about 150 milligram per deciliter triglycerides. Now, we took a deeper dive at the on the lipid fraction, and did a lipidomic analysis. And what you see here is the triple combination in the low dose arm caused a significant decrease in triglycerides, no surprise there from the preceding slide, but also produced a significant decrease in multiple bioactive lipids such as ceramides and sphingomyelins. And the higher dose caused further reductions in these glycerophospholipid species, as well as in DAG or diglycerides. Now this is a signature that is especially associated with the progression of non-alcoholic steatohepatitis. So we took a look at subjects who had high ALT, and if you look here, you see that this is the high ALT group, and this is the low ALT group. The dividing line was 50. Half the subjects were over 50, half were below 50. And what you see in the high ALT arm is virtually no change in the placebo group, but the low dose and especially the high dose group exhibited significant reductions in multiple bioactive lipid species, while the low ALT group, there's just not a whole lot going on there. So we are seeing a decrease in a lipidomic signature of NASH progression in individuals with some evidence of hepatic impairment. And to support that, this slide shows a change in liver fat, which is measured as MRI proton-derived fat fraction. And you can see that the high dose causes a nice reduction in liver fat, roughly a 15% decrease on a relative basis. So this leads us to propose that targeting energy sensing with this triple combination can successfully address multiple cardiometabolic risk factors: obesity, high blood pressure, impaired glucose tolerance, dyslipidemia, NAFLD and NASH, possibly others that we have not yet assessed. So we wanted to get a better handle on that. So to get a better handle on that, we asked the question, rather than looking at comorbidity one at a time, let's look at them collectively. So in this slide, we're looking at those individuals who lost weight and improved at least one comorbidity. And what you can see is the higher dose group, 50% of the subjects lost weight and improved at least one comorbidity. When you go to two comorbidities, that fraction does not change. And when you go to three comorbidities, you see the placebo group drops out altogether, but that 50% fraction only degrades to about 45 or 46%. And even if you ask about four comorbidities, you still have about a quarter of the subjects losing weight and dropping four comorbidity and reducing significantly four comorbidities. So this, I think, I'm going to go back a slide, lends credence to this notion that we are addressing multimorbidity with a single combination. Now, in Zan's kind introduction, he mentioned aging and lifespan, so I couldn't resist inserting just a little bit of information on that. We are not so bold as to propose that we currently have a drug that targets aging per se, but I do want to point out that we have begun to explore this notion using a variety of combinations in the simple organism C. elegans. And we've done several experiments. This shows that we can extend lifespan, both medium and maximal lifespan in C. elegans using the resveratrol leucine combination I mentioned earlier. We get comparable effects of leucine, of the leucine metformin combination. And what's interesting is if we manipulate the environment of these animals to stress them and reduce their lifespan, and we've done that in a couple of ways. One is to put them in a high glucose environment, which reduces lifespan. We can rescue that reduction in lifespan with these combinations, especially the leucine metformin combination. Likewise, these combinations rescue the reduced lifespan that is experienced in response to excessive oxidative stress. And we use paraquat to induce excessive oxidative stress, and we can rescue that with these combinations. So putting it all together, and forgive the simplistic cartoon, or please forgive the simplistic cartoon, putting it all together, we are proposing that while there are multiple expressions of the diseases of overnutrition and aging, and it's tempting to look at them, as shown here on this tree, as distinct entities that may be related to each other, we propose instead that one way of addressing them, they all have a shared feature of impaired energy sensing. There may be, almost certainly are, other modulating factors, but they all have a shared feature of impaired energy sensing as a root cause. And by addressing the roots and generating, if you will, healthy roots, we can generate healthy branches. So, to summarize, we've shown that leucine activation of SIRT1 enables synergy with other drugs that sit along the SIRT1 AMPK pathway. And we are working with some others that are further downstream and show similar effects. and that we can extend lifespan in model systems. And so targeting these energy sensing systems allows us to address multi-morbidity characteristic of metabesity. Leucine, metformin, sildenafil, this triple combination of leucine, metformin, and a very low dose of sildenafil effectively targets obesity and also targets the associated cardiometabolic risk factors. And with that, I will take. And I'm looking to see if there are any questions. I don't see anything in the chat box. If you have questions, would you please type them in the chat box?
Right. Well,
Oh, there they are.
Yeah, there they are. And while we're waiting for those questions to come in, and probably we can take them as they come up verbally. But Michael, what a wonderful tour of your, I would say, almost unprecedented findings in terms of identifying combinations of already approved agents and or a nutrient, which used in combination can affect multiple very important endpoints, and therefore has the prospects for being a metabesity therapy. And I see Todd and Art have questions. Todd, let's start with yours. What is the MOA?
Yeah, basically
Todd, out in the Bay Area.
What I'm interested in is what's the basis of the synergism, I guess? So, does leucine effect by itself have no effect? It really requires both of these things. Do you understand why it works like that?
Oh, it's a great question. Let me go back to one of my slides. I'll go back to a couple of slides, if I may. So we are counting on leucine to prime the pump, as it were, to allow for SIRT1 activation at ambient NAD concentration. That does have some effect. I won't say that it has no effect. It especially has effect in vitro, and it has what I would call modest effects in vivo for some of our variables, but not sufficient to actively treat disease. And so what we're looking for is a second or third co-activating compound. And so going back to, here we go, going back to this slide, we generate synergy, and we first showed it in vitro, and then we showed it in animals, that when we use concentrations of metformin that have minimal, if any effects independently and combine them with leucine, we get an overall increase in effect. Why? We have leucine activating SIRT1, we have metformin having an effect on AMPK and also having an effect on SIRT1, and you get an amplification. And when you add eNOS, or in fact, we've done it with nitric oxide donors as well, and with NO donors, you do get a synergy. So we really are counting on the interaction of the pathway. And I want to point out that there are other ways of activating SIRT1. You can try treating it like a light switch and putting it in the permanent on position with various activators, or through transgenics in a mouse or a rat model. We prefer a system where we can modulate. That is, SIRT1 can respond homeostatically, even with activation by leucine, metformin, and sildenafil. It can still respond to other external stimuli, either ratcheting up a little or down a little, as opposed to a powerful on switch. Does that answer your question?
Yeah, I understand that it's empirical, and the empirical evidence.
And thanks for that, Todd. And I apologize for overlooking Bart Van Schuren's question. Bart is in Belgium and a endocrinologist and also a representative to EMA. So, Bart,
can you hear me?
We hear you. We get a little feedback, but why don't you try to ask your question?
All right, the question is just the dose selection, because I don't really understand why such a low doses of eNOS activator were used.
Very good question, and thank you for asking, because I should have said so. The reason is, at this concentration, we are able to generate significant increases in nitric oxide without significantly inhibiting PDE5. We really are reframing sildenafil as a nitric oxide or as an eNOS activator, not as a PDE5 inhibitor. And at this concentration, we can get that. Now, I can offer a fairly complex explanation that would be tough without a slide, but once you introduce PDE5 inhibition and you start increasing substantially the cyclic GMP that results at higher concentrations of cyclic GMP, we actually attenuate the effects of this combination because it actually feeds back negatively on AMP. So what we're looking for is to activate eNOS modestly without an excessive cyclic GMP response.
And Bart, thank you for that important question. It's a reminder of an equally important feature of this platform, in that in the combination of approaches that Michael has put together, he's using much lower therapeutic concentration than would currently be used for the approved indication. And that is essential for having a product that would be used as a prevention in a much larger population. And by extension of that logic would justify the considerable investment that would be necessary to put on a trial to actually demonstrate these kind of long-term outcomes. And NASH is just a sort of early example of the very large and long trial that is required to show a clinical benefit, up to 3 years, and even that only provides a kind of provisional readout. And so it's just as important to have a product that's got an attractive safety profile as it is to have one that has efficacy. Sorry for that editorial comment. Let's go to Art Santora, ex-Merck or Merck alumnus and a colleague who led me from NIH up to FDA. He was at FDA before he went up to Merck. I followed in his footsteps as far as FDA. So Art, go ahead.
Hi, can you hear me? Oh, good. I don't know if I unmuted myself. I would mention that Michael and I were colleagues at Wayne State many, many years ago. So it's good to hear from you again, Mike. Great presentation. So the question, I don't know if you can read it, it's basically just asking whether in your study 0200-001, was there a correlation between the weight loss achieved and the metabolic changes achieved in, let's say, the patients who were in the high-dose group, sildenafil high-dose group? I would expect yes, but I just wondered.
Well, you would expect yes, and I would have shared that expectation, but sometimes data defy expectations. And this is one of those times. So the blood pressure change, for example, preceded the weight loss. We were able to see the change in blood pressure within 4 weeks, which was our first measurement point, before there was any significant change in weight. And there was actually not a significant relationship between them. Likewise, if we look at the change in liver fat, there was absolutely no correlation between the change in weight and the change in liver fat. There was a weak association with triglycerides.
Yeah, thanks. I'm sure you would have shown it if you had it, but did the weight loss That's a new study. That's a 002, or maybe another one. Did the weight loss continue beyond week 16? I know this was only a 16-week study, so you're limited. But
So, Art, if you wait several months, and we'll be able to answer that question for you. We have an ongoing weight management study currently, and we expect to read out later this year.
yeah, that would have been my guess. Thanks for confirming. Thanks.
I'd also had a follow-up question, Michael, and he simply asked, have you looked at other branched-chain amino acids?
Yes, we have, and thank you for asking that. These effects of leucine are unique. We have tested each of the other branched chain amino acids and as controls multiple non-branched chain amino acids. And this is a unique effect of leucine. Neither isoleucine nor valine exert these effects in terms of SIRT1. On the other hand, high doses of the other two branched chain amino acids, as well as some other amino acids, do have the potential to have the opposite effect, that is, to affect mTOR. But no, they don't affect SIRT1.
And we have a call from my friend in Philadelphia, or outside of it, Andrew Room, who has a more of a regulatory question. But go ahead and ask it, Andrew. Let's see, Andrew, if you are, I'm trying to see if you're muted. I don't know what happened to Andrew, but come back if you can, Andrew. Anyway, your question, Andrew's question, is impaired energy sensing likely to be a regulatory acceptable indication? Michael, you might take a stab at that.
Well, what I will say is whatever I say, I'm hoping Zan will come right behind me because he's got he may have a longer view. My answer is not yet, certainly not presently. I think presently we have to focus on the resulting disease states, diabetes, NASH, hypertension, obesity, etc., as indications. Long view, I can say I hope so, but I will ask Zan to probabilize the likelihood.
Well, I do too. It may not be so much a functional defined indication, but at least we do need regulatory indications that relate to prevention, that relate to modifying the aging process. And that is an area of a lot of thought, not so much action at FDA, but there are mincing steps being taken. For example, under consideration right now is a type 2 diabetes prevention indication for metformin. That's already been awarded, by the way, by multiple authorities, including, let's say the UK, for example. So that's a very easy low-hanging fruit. It's going to be much harder to get to substantial indications that would justify treating large numbers of people to prevent diseases or infirmity.
Would love to get there.
And I think you're helping us to get there, Michael, because as you know, you heard a talk by Nir Barzilay in Boston recently, where we were having a more limited discussion about diabetes therapeutic development. But Nir also spoke at the London conference that Thomas mentioned, and Nir is leading this prevention trial using metformin aimed at a composite of age-related comorbidities, which is essentially the first metabesity trial, a trial to show that these comorbidities can be prevented in aggregate. It's a very exciting step being taken. Unfortunately, even though I approved metformin at FDA, and it's been sort of my a favorite subject of mine, I don't think it's a particularly potent agent to be going up against these comorbidities. I think what Michael is showing us here has much greater promise in just that trial design that NIR has come up with. We certainly hope that that trial is going to be positive. It will at least set a precedent for a design and an execution of a trial that is aimed at this very novel therapeutic indication.
Absolutely. Nonetheless, I very much look forward to seeing NIR's TAME study progress.
And Todd, you're coming up with great questions. You have one about NAD levels. This has been another area of a lot of interest. And in fact, you can buy a product today that increases supposedly intracellular NAD tone. And that does have a story for increasing lifespan. So Todd, are you still there? And would you like to flesh out your question?
I'm just curious. I keep getting ads on my Facebook page about this, I haven't looked into it myself. So it looks kind of like some similar ideas, but a different mechanism. So I thought I'd ask Michael to comment on the whole idea of increasing NAD levels and the best way to do that if it is a good idea.
So I'm familiar with the ads that you're getting. It's from Elysium, I think is the name of the company, although there are others that also sell the same product. It's nicotinamide riboside. And actually, they published a paper, it was either earlier this year or very late last year, I've forgotten which now, in Aging Cell, showing clinical trial data that they were able to successfully elevate NAD levels. I mean, that was the primary purpose of the trial. It's really not a different mechanism. They're counting on NAD to activate SIRT1. And in fact, we've done some work with NAD donors, including nicotinic acid, to good effect. We've managed to show lovely reductions in lipids, very nice synergy when we combine leucine with an NAD donor. I don't know, however, what the overall systemic effect is of providing an NAD donor long term. I find the science excellent. I find the therapeutic potential of interest, but from a safety perspective, in my mind, it's a bit of a black box. It's a little different than combining well-known, well-characterized compounds with long-term safety records. And the data I've shown you, of course, has sildenafil, leucine, and metformin. It's other than a little GI distress from metformin for some individuals, it's really going to be hard to point to much in the way of adverse effects. I'm not suggesting that nicotinamide riboside has them, but anytime you have a potent activator of biological systems, you certainly want to look for them. And I just don't know the answer to that question long term.
I think is the mechanism that leucine does this. Is this by stimulating AMPK primarily, do you think? Because I do believe that if you change the NAD to NADH ratio in favor of NAD, that also can stimulate AMPK.
Well, so the answer is yes, kind of. The primary effect of leucine is on SIRT1. But once you activate SIRT1, SIRT1 acts on AMPK through its upstream activator LKB1. And so SIRT1 does have a meaningful effect. And so, yes, in our systems, we do show increases in AMPK, but they're primarily dependent on SIRT1.
you very much.
Great discussion. Thanks again for that question, Todd. It also points out a real challenge for us, for our society, in how do we get evidence for products that could have a substantial effect on, for example, the aging process. And in this case, we already have a marketed product. It's not a drug. It's a nutraceutical that is marketed under a completely different regulatory pathway, one that doesn't require evidence of clinical benefits, and likewise does not allow disease claims. But Elysium is off and running with a product, and it may well provide substantial benefits, but will that company have the means to conduct long-term trials to show that the product works? What will the incentives be for showing such evidence? I think it's unlikely, short of NIH doing such a trial, that we'll get that evidence. And so we could have decades of using a product with very limited evidence that that's doing any good, or that there may not be some safety issues to it. So that's food for thought about the sort of alternate marketing pathways that are frequently used when it comes to the so-called anti-aging therapies that are out and about.
I agree.
I'm looking to see if we've missed any questions. Art, I see you followed up with comments about FDA or potential FDA indications. Would you like to parse that a bit?
Yeah, so somebody mentioned probably about 10 minutes ago about, pathways to approval. My guess is if you could show a very nice anti-obesity weight loss effect, that'd be enough to get it by, given that the safety and efficacy were fine. NASH could follow. I don't know. I mean, maybe it's preliminary to ask Michael what he intends to go for first, but I think there's a pretty big potential market in obesity, particularly if a drug actually works with minimal side effects.
So, Art, I'm happy to answer that question. We currently have an open IND in obesity and another one in NASH, and we're working towards both endpoints. We will have the rest of our phase 2 data in obesity later this year, and so I suspect that is where we will. I think our strategy very much mirrors what you're describing.
And also to add, what is almost a quirk these days, our outcome-driven therapeutic development environment, is that at FDA, probably the lowest hanging fruit for this therapy would be a hypertension indication. The effect size that Michael has seen would certainly be over the threshold needed to say that the therapy is effective, and there is no requirement for an outcome trial. Blood pressure medicines are still approved on the basis of the effect on blood pressure, so it's another potentially low-hanging fruit.
Mm-hmm. And that is one that
Zam, this is Thomas. I'm probably opening up a whole can of worms here, but I'm struck by emerging science that looks at molecular pathways and intervening there, and they're having interesting results, beneficial results in multiple diseases and conditions. I just came from the Defined Health Cancer Progress meeting earlier this week in New York, and there was a very exciting presentation about the future of cancer care, where you would hit them early and aggressively. You would sequence every tumor. The statement was made, if you're not sequencing within 5 years, it'll be malpractice. And then you draw upon your algorithmic sheet for various molecular pathway targeting to put together a cocktail, because cancer is so slippery and can evolve out of a particular medicinal route, and to hit them early and often. And for that kind of an approach to work, it's all kind of both off-label and off guidelines, which are maybe 5 or 10 years behind. And yet, this may have the greatest practical effect for. One doctor was reporting 50 to 80% response rates. So I wonder if you could comment on the conceptual possibility of the FDA looking at molecular pathways or functional intervention. The promotion for this talk by Michael referred to Nick Lane saying, if you could just target free radical leakage from mitochondria, you might be able to address 10 diseases, non-communicable diseases of aging. Zan, is that just too hot a potato to talk about?
Well, I think conceptually it's very appropriate. Of course, there's some major distinctions to be made between targeting existing cancer and what is being entertained here is a prevention approach. But by the way, cancer prevention is very much in this constellation of metabesity. Michael is obviously not approaching cancer as a therapeutic proposition for obvious reasons, but there is good evidence that even metformin, for example, reduces cancer risk. So we should make the distinction between prevention and disease treatment, but part of the paradigm that you described could be a metabolic sort of background therapy that would synergize with these heavy hammers that are being used, and though effective, are also they have a price to pay, or we pay a price to use them. The standpoint of safety.
Well, we've reached the noon hour, and I just found this fascinating. I want to thank everyone for coming, and I want to announce that our next public webinar will be on Friday, June 8th, where Bart van der Schoen will speak on European regulation of medical products, and in July we'll have an expert in the Japanese regulation of medical products. So we'll continue this series. I hope you find these interesting and useful, and we'll see you next time.